Hi guys, today we will be talking about energy, enzymes, and metabolism. This is kind of a big chapter. So, um, energy essentially flows through an ecosystem. And so, a big deal with energy is energy transformations. We know we're living organisms and we really need to be able to process energy and also feed our cells essentially so that we can continue to do things that require energy, like active transport type things. So, the term energy itself is the ability to do work or bring about change. And so, energy itself flows through that ecosystem. It does not cycle. You don't get to use it and then make more essentially without an input of energy. And so we talk constantly about kinetic energy versus potential energy. So kinetic energy is literally mechanical energy or energy of motion, whereas potential energy is energy that is stored and could be used in the future. So that's like chemical energy. You're making essentially, if you're a plant, you are able to generate or make your own potential or stored energy when you're doing photosynthesis and you're making your own sugars. This is a representative of your textbook's example of flowing through an energy system, through an ecosystem with energy. So you can see energy comes in through the sun, it's taken in by photosynthesizers, and of course transferred by organisms who eat photosynthesizers and so on and so forth. And so every time you see that energy transformation, you do have some energy lost as heat. So with that, we have to talk a little bit about the laws of thermodynamics. And we just talk about the first and second laws of thermodynamics because they're applicable to the way we're looking at energy transformations. And so the first law of thermodynamics is the law of conservation of energy. So energy can't be created or destroyed, but it can be changed from one form to another. So like potential energy can be broken down and processed or converted into mechanical energy. The second law of thermodynamics is the law of entropy. And so with this law, when you do see that energy change from one form to another, there is always a loss of usable energy. It's not a 100% efficient task to change from one form of energy to another. And so that wasted energy that you're not able to use generally is lost as heat and goes to increase disorder, like in the environment. So with metabolic reactions and energy transformations, basically you're looking at the metabolism of a cell. So metabolism is basically the total chemical reactions in a cell. And so with metabolic reactions, you have reactants that participate in a reaction, and then you have products that form as a result of a reaction. So reactions can release energy or require energy. So those that release energy, or ATP, are exergonic reactions. And those that require energy are called endergonic reactions. So cells generally use energy by unlocking stored glucose that's being saved up in a cell. So glucose essentially gets broken down. Only about 39% of the energy gets converted. And so we have to break down that glucose in order to make ATP. And ATP is essentially adenosine triphosphate. So it's a really high energy compound that's used to drive metabolism. Um, and cells must receive a constant supply of food to continue to make ATP. ATP is continuously made and broken down all through our days and nights. Like as long as we're living, we're consistently needing ATP input and we're consistently breaking it down. We don't store ATP in large ways. Like we can store our energy reserves like glucose and things like that. So ATP is basically synonymous for energy for cells. So it's adenosine triphosphate, and it's constantly being generated from ADP, or adenosine diphosphate. And so when you think about the words triphosphate or diphosphate, you can imagine that what you're seeing is actually adenine, ribose, and then together, remember adenine is one of those nitrogenous bases kind of thing, ribose is a sugar, so you've gotten adenine and ribose together, which makes adenosine, and then you have three phosphate groups, and that bond between the second and the third, that last phosphate, is kind of wily, and so breaking off that kind of unstable, wily bond actually releases energy, and that energy is what we use to power all of those processes in our cells. That's probably why ATP is called commonly the energy currency of the cell, because energy from that broken bond is sufficient enough to drive the cell's metabolic reactions. This is a reflection of the ATP cycle. So basically it just says that we're constantly using ATP, and then we're constantly having to make more ATP by joining that third phosphate to ADP. So ATP in general does chemical work. It makes proteins and lipids. It's responsible for helping with transport by helping pump materials across the membrane, like we saw in the sodium potassium pump. And then also even mechanical, so muscle contraction, cilia, flagella movement, everything that we need to do. Enzymes themselves play a huge role in metabolic processes and energy transformations, like huge role. You can almost say that there's enzymes almost help with almost every reaction that happens in a cell. So they're known as proteins. So most enzymes are proteins, and they are necessary for increasing the rate of chemical reactions. They do a lot more than that, though. Enzymes themselves do a lot more than just catalyze or increase the rate of chemical reactions. Most enzymes are generally kind of easy to determine when you're like looking at a word and it ends in ace. It's almost always an enzyme, at least in science. So lactase, catalase, lipase, cellulase, those are all different types of enzymes. And they essentially work to bring things into contact with one another that might not happen otherwise. And the speed of the reactions that enzymes work are basically based on enzyme and substrate amount, the temperature of the location where those enzymes are being used, and even the timing can affect the speed on enzymatic use. So enzymes help to function as catalysts that bind to substrates. So each enzyme is responsible for accelerating a very specific reaction. And so reactions in a metabolic pathway actually require a specific enzyme to work. You're not going to get the end product unless all the enzymes in that metabolic pathway are present and functional. So you can't actually get to B without an enzyme present. You can't get to C without an enzyme present. So essentially, enzymes work by binding to a substrate, and you actually are able to form a product. And so they make what's called a enzyme substrate complex. And so there's a specific site called the active site where that substrate will bind with. So then the active site complexes or binds with the substrate, and that causes the active site to change shape, and the shape change will force substrates together, initiating a bond. So not every enzyme will join together a substrate molecule, two substrate molecules to make one product. Sometimes you will have like a single substrate and that gets broken apart into two products. And when you have that, that's called degradation. So you're taking one thing and you're making two products. Now, synthesis is when you're combining two substrate molecules and you join together to release one product. And this comes from your textbook, but it kind of shows how degradation might have. You might have one single substrate and you might break it down into two products, or you might have two substrates and you might have your enzyme complex and then release one product. So enzymes work, and they actually work to do so many things. Like I call them like the Prius of the metabolic world because they help reactions happen more quickly and more efficiently, and they bring the substrates or the things that you're working with, they bring molecules together. And initially, you might not see molecules interacting with one another, but enzymes can help make that happen. But enzymes work at particular speed, and the speed at which enzymes work are affected by some different factors. And when we do our enzyme lab, what you're going to notice is that we actually get to test all of these factors. So one thing that affects enzymatic speed is substrate concentration. So if you have a lot more substrate, of course, then there's going to be more frequent collisions between the substrate and the enzyme, and you're going to see that enzyme work a lot faster. Temperature can even increase substrate activity. Warmer temperatures cause more collisions between the enzyme and the substrate. However, if it's too hot, a really hot temperature can denature and destroy an enzyme. And so denature means that because remember, enzymes are proteins, and proteins have to be shaped very specifically, and they have to be folded just so to function. And so if you overheat that enzyme, then it's not going to be able to change shape anymore, and that's when you're going to see denaturing. We're going to denature the heck out of some enzymes in lab. Then also something that affects enzymatic speed is pH. And so not to say that, oh, if it's acidic or if it's basic, an enzyme can't function there, that's not true at all. Most enzymes are optimized for a specific pH. So there's lots and lots of enzyme activity in our stomachs, right? But our stomachs are super acidic. But that's okay because those enzymes that work there are specifically designed to function in that acidic environment. And if you took them out and put them somewhere more basic, then, I mean, you wouldn't even have that function work at all. Like the enzyme wouldn't work in a more basic scenario. Some cats actually have a mutation that causes an enzyme to be active only at cooler body temperatures. And so only certain regions of that body are dark. And so areas will actually darken with age and decrease circulation. Cells can also regulate the presence or the absence and even the concentration of an enzyme. It's a cell's job to do that. Enzymes also have what are called enzyme cofactors. So those are molecules that are required to activate an enzyme, and they can be metal ions or they can be coenzymes. So some really important coenzymes include NADP, NAD, FAD. Those are all super important, and we're going to talk about them very, very heavily within the next couple chapters because they tend to act like batteries and they carry hydrogen and electrons as well, and both of those are needed for certain metabolic reactions, specifically like photosynthesis and cell respiration. So those coenzymes are non-protein organic molecules that help the action of the enzyme by binding to it. So here's a cofactor showing you the active site. And then you can see how that helps a substrate bind in that active site. So sometimes reactions need stopped or slowed down. Like if you have made enough product, you don't want any more product, then enzymes can be inhibited. And so there are such things as non-competitive or competitive inhibition. That's two ways that enzymes can be inhibited. And these are reversible inhibitions. Like these aren't non-reversible. Like say you don't need so much product for now, but you might eventually need that product again. So this enzyme activity isn't stopped forever. It's just stopped while you have these inhibitors in place. So cells regulate that, not us. Like we aren't like, hey, I think I'm done with the lactase now and I'm no longer going to be breaking down lactose, so stop cells. No, your cells just do that. So a non-competitive inhibitor would be where you have an inhibitor that binds at what's called an allosteric site. And when that binds there, then the active site where normally your substrate would bind on that enzyme changes shape. And so this no longer fits there, right? So it's non-competitive. You don't see like what happens with a competitive inhibitor is that you'll have what happens with a non-competitive or what happens with a competitive inhibitor, both the active site and both the substrate and the inhibitor are all vying for the same active site. So both of those conditions, like if the competitive inhibitor binds at the active site instead of the substrate, then you're not going to make a product. But if the substrate binds at the site, then you'll get your product, or they're both vying for that active site. So these are reversible enzyme inhibition. So essentially, if you happen to need that product again, it's possible that you are able to do that. It's possible that you're able to see that enzyme activity begin again. Now, there are non-reversible enzyme inhibitors, and those are just called poisons. So poisons irreversibly inhibit an enzyme, and they can lead to damage or death, and death. So like one of the really well-known irreversible enzyme inhibitors is called cyanide. So cyanide essentially binds with an enzyme required for ATP production, and if you can't make ATP, you die. Serine is a really good inhibitor that binds itself to an enzyme located at the neuromuscular junction, and then you can't, you can't function, you can't move. Warfarin is actually a well-known medical inhibitor that's used for, like if you've ever had heart surgery, you may be prescribed warfarin in low doses and medically regulated doses to prevent you from blood clotting. So it's very common for, it makes it impossible to clot your blood. But like when you're prescribed medically regulated warfarin, typically known as like Coumadin or things like that, those help prevent blood clots. So we've talked about the flow of energy, and we've talked about how enzymes help metabolic reactions. One of the most important reactions that we talk about as we talk about energy transformations, and that's what we're going into, are known as redox reactions. And so with a redox reaction or an oxidation reduction reaction, all it is, and it sounds super big and hairy and scary, but all a oxidation reduction reaction is where electrons pass from one molecule to another. That's it. It's a super hairy word for one molecule passing along an electron. So oxidation, the term oxidation, is just a molecule that's losing an electron. Reduction is a molecule that's gaining an electron, and this happens at the same time. One molecule is like, hey, I've got this electron. Not really. Really, an electron moves from one molecule to another. So the one that lost the electron has been oxidized. The one that gained an electron has been reduced. And the way that I remember oxidation and reduction is the term oil rig. So oxidation is losing an electron, where reduction is gaining an electron. Oil rig. So two of the most important redox reactions that we talk about in class are, of course, cell respiration and photosynthesis. So cell respiration is basically breaking down a glucose molecule to access that stored chemical energy, and that happens in the mitochondria. And then photosynthesis is another important redox reaction that uses solar energy to basically activate and store chemical energy in the form of glucose, and that happens in the chloroplasts. So if you look, this is super critical, and when I was in college, it took me about 10 eureka moments before I got over the fact that photosynthesis and cell respiration as a process are literally backwards from one another. So photosynthesis, you're building a glucose molecule. Cell respiration, you're breaking down a glucose molecule. That was a wow moment for me, I'm not gonna lie. So if you memorize one, and you should, you will essentially have both of them memorized. So one of the important things that we see often, and we talk about often when we're talking about these redox reactions, are what's called an electron transport chain. So it's essentially, remember we talked about plasma membranes and we talked about double membranes. Well, in the mitochondria and in the chloroplast, there are these double membranes that have membrane-bound carrier proteins. So they are physically arranged in an ordered series, and they start with high energy electrons that get passed from one protein carrier to another. So it's almost like a weird electron game of hot potato, because obviously the most energy that you have from electrons are seen in that first carrier bounce, go from one carrier to another. That electron energy, and we said electrons, we said way back in chapter 2 that electrons have a ton of potential energy because they are essentially able to, I mean, they have to have that energy because we keep them in those electron shells. And so it takes energy to keep them there. So we like to utilize and harness that electron energy to pump hydrogen ions to one side of the membrane, and that will create an electrochemical gradient across that membrane. And then we use that gradient to push those hydrogen ions from areas of high concentration to low concentration. And so because we're moving hydrogen ions from high concentration to low concentration, it's like osmosis, but with a chemical. So we call it chemiosmosis. And what you end up with is low energy electrons but you were able to make high energy ATP. This image comes from your textbook, and it does kind of show how high energy electrons are passed from one carrier protein to another down the electron transport chain so that you can generate ATP. This shows you chemiosmosis, and you're able to pass those electrons from one protein to the other.